July 28, 1996. Spectators line the Columbia River for a boat race in Kennewick, Washington. Two college students wade into the shallows and feel something hard beneath their feet. A human skull, 9,000 years old. Its strange features would divide the scientific world, pit researchers against Native American tribes in court, and ignite a decades-long mystery that only ancient DNA could finally solve. On July 28, 1996, the Columbia River buzzed with the noise of hydroplane engines and the cheers of race fans. Amid the excitement, Will Thomas and David Deasy, two college students, waded into the shallows near Columbia Park. Their feet struck something unexpected, a hard object half buried in the silty riverbed. It wasn't a stone. When they pulled it free, the realization hit. They were holding a human skull, weathered and ancient, not the remains of any recent tragedy. The skull was placed in a 5-gallon plastic bucket and handed off to a nearby police officer. From there, it traveled quickly to the office of Benton County Coroner Floyd Johnson. The coroner, faced with a skull that didn't match any missing person or recent case, called in James Chatters, a local forensic anthropologist known for his expertise with skeletal remains. That same evening, Chatters examined the skull at his home in Richland. He noted the extreme wear on the teeth, the bone's mineralized texture, and the absence of modern dental work. These clues pointed to a much older origin than anyone had initially guessed. Chatters wasted no time. He notified the Washington State Archaeologist and the University of Washington's Department of Anthropology. Within days, a team assembled at the riverbank. What began as a chance find quickly became a full-scale recovery operation. The excavation revealed far more than a single skull. Layer by layer, the team uncovered a nearly complete human skeleton, over 90% intact, embedded in sand and gravel. Every bone was carefully mapped, labeled, and wrapped in acid-free tissue for transport. The remains were transferred under strict chain of custody to the University of Washington's Anthropology collections, ensuring their preservation for further study. The next step was to determine just how old these bones were. A small sample of rib collagen was sent for radiocarbon dating. The results, when they arrived, stunned the scientific community. The individual had died approximately 9,000 years ago, with a margin of error of just over a century. This made the skeleton not only one of the oldest ever found in North America, but also one of the most complete. The find was immediately recognized as a scientific treasure, promising new insights into the early history of the continent. As word spread, local headlines described the mysterious skull discovered by two spectators. Television crews filmed the riverbank, capturing the sense of wonder and confusion that hung over the site. In labs and universities across the country, archaeologists and anthropologists began to speculate about the origins of this ancient individual. The stage was set for a scientific investigation that would soon spiral into controversy, pitting competing theories and worldviews against each other. In the months after the bones arrived at the University of Washington, the laboratory became a battleground for dueling interpretations. R.J. Crawford, a physical anthropologist, was among the first to publish craniometric data on the skull. Crawford's measurements emphasized the skull's long, narrow shape, dolichocephaly, and a pronounced brow ridge. He compared these traits to European Upper Paleolithic specimens, arguing that the Kennewick skull fell outside the range of known Native American crania. Crawford's data sheets, filled with caliper readings and cranial index calculations, suggested a possible link to ancient Europeans, a claim that quickly drew headlines and fueled public speculation. M.R. Berin and his team pushed back with equal conviction. They pointed to features that told a different story: shovel-shaped incisors, robust cheekbones, and a cranial index that overlapped with early Holocene Ainu and Jomon populations from northern Japan. According to Berin, the dental and facial traits aligned more closely with Northeast Asian groups, consistent with migration across Beringia. Both sides marshaled comparative datasets, Crawford drawing from 19th century European collections, Berin relying on Asian Holocene series. Yet neither could escape the methodological pitfalls. The skull had been compressed by river sediment, possibly distorting key measurements. Reference samples were limited, and the distinctions between populations blurred by thousands of years of human movement. While scientists clashed over calipers and data points, the stakes intensified legally. Under the Native American Graves Protection and Repatriation Act, or NAGPRE, 4 Columbia Plateau tribes, the Umatilla, Yakima, Nez Perce, and Colville, filed a joint claim for the remains. They cited oral histories, geographic proximity, and cultural continuity, insisting the bones belonged to their ancestors. The case moved rapidly into federal court. In 1999, the U.S. District Court denied the tribal claim, holding that the scientific evidence did not establish a direct cultural link. The decision allowed research to continue, but the tribes appealed. For years, the case wound its way through the courts, with scientists and tribal leaders testifying on opposite sides of the divide. In 2004, the Ninth Circuit upheld the denial, citing a lack of definitive affiliation and reinforcing the primacy of scientific study over repatriation. As the legal battle dragged on, the remains sat in storage, locked in a legal limbo. Each new court ruling raised the stakes, turning a disagreement over skull shape into a test case for how America handles its oldest human ancestors. The controversy exposed the limits of traditional physical anthropology, and the courtroom became the new arena for a debate that science alone could not settle. The question of who Kennewick Man was and who had the right to decide remained unresolved, caught between the tools of the lab and the demands of the law. For years, the debate over Kennewick Man's ancestry circled around calipers and data sheets, but cracks in the old approach grew impossible to ignore. In 2002, anthropologist M.D. Proofer published a pointed critique, arguing that the skull's shape had been warped by centuries of river sediment and burial pressure. Measurements that seemed to hint at distant origins could just as easily be artifacts of deformation. Reference collections, too, were limited, often drawn from modern or historic populations that bore little resemblance to people living 9,000 years ago. The tools of classic craniometry, once seen as objective, now looked more like a lens clouded by time and circumstance. As the legal and scientific stalemate dragged on, a new path opened in the laboratory. Eske Willerslev, a Danish geneticist renowned for extracting ancient DNA from some of the world's oldest remains, received permission to attempt what earlier generations could not, sequencing the genome of the ancient one. The stakes were high. Any hint of modern contamination could compromise the results, so every step demanded precision. The team worked in a clean room at the Max Planck Institute, where the air was filtered, surfaces were sterilized nightly with UV light, and researchers wore full bodysuits. Before sampling, the outer surface of the selected bone fragment was scrubbed with bleach and blasted with ultraviolet to destroy stray DNA. Only then did they drill into the untouched inner bone, collecting powder for extraction. The DNA was coaxed out with a silica column, a method that binds even the tiniest fragments. Initial sequencing on the Illumina HiSec 2000 produced over 1 billion raw reads, but most were environmental debris. To enrich for the ancient human genome, the team used a targeted SNP capture array, zeroing in on over 1 million positions known to vary among global populations. Throughout, contamination checks showed less than 1% modern DNA, an unusually clean result for a specimen this old. After months of painstaking work, Willeslev's team finally had enough authentic genetic data to run ancestry analyses. The moment of truth had arrived, promising answers where calipers had failed. The results from the genome analysis arrived with a clarity that decades of argument had failed to provide. Principal component analysis placed Kennewick Man squarely among present-day Native American populations of the Pacific Northwest. The closest genetic matches were the Colville, Nez Perce, and Yakima tribes, groups whose oral histories had long claimed ancestral ties to the ancient one. Admixture modeling confirmed a dominant Native American component, with no significant signal from European or East Asian populations. Statistical tests, including F4 statistics, ruled out any excess genetic affinity with non-native groups. The mitochondrial D belonged to haplogroup X2A, a lineage found almost exclusively among indigenous peoples of North America. When the findings were published in Nature in 2015, the scientific debate that had raged for nearly 20 years came to an abrupt end. Esker Villerslev, who led the genetic team, called the result unambiguous. For tribal elders, the DNA evidence was not a revelation, but a confirmation. Statements from the confederated tribes of the Umatilla and Yakama Nation echoed a simple truth. Science now confirms what our ancestors have always known: the remains belong to our people. The legal and cultural consequences were immediate. Armed with genetic proof, the Army Corps of Engineers authorized the transfer of the remains. In February 2017, tribal representatives gathered for a private reburial ceremony near the Columbia River. Elders from the Colville, Nez Perce, Umatilla, and Yakama nations presided, fulfilling a promise that had lingered since 1996. The following year, the National Park Service updated its guidelines, requiring that ancient DNA be considered in future repatriation claims. Kennewick Man's genome did more than resolve a scientific mystery. It changed federal policy and restored the ancient one to his descendants. Today, DNA evidence settles debates that once divided science, law, and culture. As genomic technology reshapes our view of the past, it also redefines identity and belonging. The search for truth now demands precision over speculation, and the story of who we are is still being written. Share your perspective below.